Today's symposium is oxygen uptake and oxygen extraction during exercise, implications for health and performance. The idea for the symposium and much of what will be presented today has evolved from a 10-year collaboration we've had with Dr. Silvia Pogliagh, who will be our third speaker, between Western, the University of Verona in Italy, and more recently, the addition of the University of Calgary. So over 10 years, we've collaborated in various projects, had students go back and forth between the two labs, and it's been a very productive international collaboration that is continuing. So much of what you'll see today evolves from that collaboration. The symposium is sponsored by COSMED. COSMED equipment was used to measure the oxygen uptake in many of the studies that are presented. Our first speaker is Dr. Juan Murias at the University of Calgary, and he'll talk about mechanisms controlling oxygen uptake during submaximal and maximal exercise using both oxygen uptake measures and near-infrared spectroscopy measures. Second speaker from the University of Western Ontario and presently in a postdoc at the Toronto General Hospital is Dr. Keir. He'll talk about oxygen uptake and NIRS derived determination of exercise intensity thresholds and their implications for exercise prescription and performance. And third and best will be Dr. Silvia Pogliagh. Should practitioners consider oxygen uptake as a vital sign? So first of all, I'll call on Dr. Murias to come forward and present on mechanisms controlling the adjustment of oxygen uptake.
Okay, hello everyone. Thanks for coming. Thanks for showing up. Thanks for your kind introduction, Don. Always a pleasure having you around and saying that the third speaker will be the best. That's the way I wanted to start. So yeah, you can come back in an hour. That's it. So as Don said, I'm going to be talking about oxygen transport during exercise and oxygen extraction and some implications about oxygen delivery. Something funny with this big room and having two screens is that I'm not going to be going back and forth with the laser. I don't want to hit anyone. So I will probably focus on this one. I'm a left-handed person, so it's more convenient for me. So I apologize for the people on this side of the crowd. So the way I'm going to be targeting my presentation today, I'm going to be talking about two different models that we have been using for the past 10 years, as Don said before. One component uses this, oops, my pointer is different. So yeah, here. So one model uses this oxygen uptake kinetics profile, and the other one is going to be using ramp incremental exercise and looking at VO2 and oxygen extraction data and what we can learn about oxygen delivery from those two data sets put together. So since I'm going to be starting with the oxygen VO2 kinetic stuff, I thought for some of the people that are not that familiar with the concept, I would put together some slides about what VO2 kinetics is or how we measure it in the lab. So basically, oxygen uptake kinetics is how the oxidative metabolism, the aerobic metabolism adjusts to a change in metabolic demand, how quickly it does it. When we measure it in the lab, we have people riding at a constant power output starting at 20 watts, and then we change the intensity in a step wave form, so instantaneous increase in metabolic demand. So to that instantaneous increase in metabolic demand, what you see is a rather exponential increase in VO2. Nothing too new about this. That response, when we measure at the level of the mouth, has three components. The phase one of the response, which is the cardiodynamic phase, and then we have a second component, which is defined as the main component of the response, and that main component is what we are going to be focusing on for this presentation, and then the third component, that during moderate intensity domain, which is what we are going to be talking about today mainly, is steady state exercise. So from a practical perspective, we'll be focusing on a number that comes from this tau value. So the smaller that number, the faster the adjustment of oxidative metabolism. And from a practical perspective, the implications of this speed of kinetics is that some people have a relatively fast adjustment, as you can see here, someone with, let's say, 25 seconds VO2 kinetics, and some people have a much slower adjustment. So we'll talk about it later, but this is typically in older people or maybe in untrained, healthy untrained people. So the implication of that is that a larger oxygen deficit and more reliance on non-oxidative sources will be associated with a slower VO2 kinetics response. So the faster it is, the less the reliance on oxidative sources. So one of the questions that has been of interest for the past 20, 30 years when talking about VO2 kinetics is what are the mechanisms that control this response? And this is a little bit of what I'm going to be describing when talking about oxygen delivery, oxygen transport, and how important it is. So that's the equation that describes oxidative phosphorylation. So these are all the things that we need for ATP to be resynthesized aerobically. And the main theories for what controls how quickly oxidative metabolism occurs are that there is an oxygen supply limitation or some intracellular components that are either making that adjustment slower or faster. And then there is people saying, well, it's a combination of both. And the reality is that, well, obviously it has to be a combination of both because we need all the substrates to make oxidative phosphorylation happen. However, the data that I'm going to present today, I hope it somehow convinces you that oxygen transport to the active tissue plays a critical role in making this adjustment faster. So a little bit of a historical overview. So I know no one can actually read those numbers, and I apologize for that. But basically, this is a study from Dr. Houston's lab, and what they did, they showed that when people were cycling in the upright position, VO2 kinetics was around 24 seconds, and then when they put people to exercise in a supine position, so in an unfavorable condition for oxygen delivery to the legs, the VO2 kinetics got slower. And then when they had people cycling or exercising in the supine position, but with lower negative pressure, so that now oxygen delivery is improved, so then the VO2 kinetics response got back to the about 24 seconds kinetics. So quite a bit of work from Dr. Houston's lab and people in that region where they were showing with different experiments like hypoxia or beta blockers to impair cardiac output or reductions in perfusion, that you could actually make VO2 kinetics slower. So that was the proof back then that, okay, oxygen delivery plays a role. But there wasn't much evidence supporting the idea that you could make VO2 kinetics faster by increasing oxygen delivery to the tissues. So then, a little bit later, these papers by Bruno Grassi were one of the strongest pieces of evidence that oxygen delivery to the tissue had very little to do with the adjustment of VO2 kinetics. And what they did, so they had a control condition. This model is in dogs. And they had a control condition where they measured VO2 kinetics in these dogs, and those are the empty circles there. And then they had an experimental condition. What they did, they improved oxygen delivery before the onset of exercise to what it could be at steady state exercise. So before even the onset of exercise, oxygen delivery or blood flow would match the oxygen transport that was expected for steady state exercise. And even though they improved that, the oxygen delivery or the oxygen kinetics remained the same. And then they did another experiment that was very interesting, where not only improved oxygen delivery, but also oxygen diffusion by giving these dogs hyperoxia. and a drug that would improve oxygen diffusion to the mitochondria, so, or offloading of hemoglobin from the red blood cells to the mitochondria, to the cell. So again, same story, no change in VO2 kinetics, and basically what they propose is that neither convective nor diffusive delivery of oxygen had anything to do with VO2 kinetics. Something important that this model was in dogs, highly oxidative animals, which had a VO2 kinetics that was already fast enough that it's very difficult to find any further changes. Once you have people or animals with fast enough VO2 kinetics, it's difficult to find further changes. And then the last study that I'm going to show you before starting with our own data, or the data that I collected in the past years, it is a study by Greg Dumanoir in Don Paterson's lab. So he was looking at the rate of VO2 kinetics, the rate of adjustment of VO2, as well as the rate of adjustment of oxygen delivery in the femoral artery. So what he found was that the tau values were the same for lean blood flow and VO2 kinetics. So the idea is, at least at the conduit artery level, oxygen delivery adjusted at the same rate as VO2. So the basic conclusion would be like, okay, it doesn't seem that oxygen delivery is playing much of a role to make VO2 kinetics faster. However, in that same study, and I wish Greg was here. Are you here, Greg? No. He's constantly accusing me of taking credit for the work he did, and I always try to acknowledge his work, but he doesn't show up. He went to the next, to the other symposium. So that's sad. Anyway, so he was integrating not only the leg blood flow and VO2 signals together, but also this signal that I'm going to be describing soon, the HHV oxygen extraction data derived by near-infrared spectroscopy. So what he found was that there was a faster adjustment of oxygen extraction for a given adjustment in VO2, which implied then that there might have been an oxygen delivery limitation because we were more reliant on oxygen extraction. So I'm going to get more in details into that soon. So before I get more into the mechanisms that control the VO2 kinetics response or oxygen delivery to the tissues, I'll say a few things about near-infrared spectroscopy. A lot of our data is based on oxygen extraction signal derived from the HHV, so I'll call it from now on oxygen extraction for simplicity in most cases. And the physics behind NIRS are complex, or at least complex enough for me. But basically what happens, there is an emitter sending near-infrared light through the tissue, and then there are receivers that pick up the light, and based on the amount of light that is reflected back in those receivers, then we can have an idea of what's happening with the oxygenation status of the tissue. And I put these pictures here because this was the first system I was using back at Western. This is the Oxyplex system that I'm currently using at University of Calgary, and Silvia is using in Verona. But now there are more portable devices. So this one, the Portamon, not only is portable and wireless, it also interfaces with your phone. So you can collect your own data while training. And then Moxie is a similar system that basically does the same thing, and it has apps so that you can download your data. And one of the interesting things is that this, oops, again, it's not my pointer, it's different. This system here costs about $60,000. This portable system here costs about $15,000, and this one here costs only $1,000. And they are trying to target this system more and more as a training, not only research, but also as a training device that people could use on a daily basis. And Dan will talk a little bit more about that later, the implications of some NIRS data. One reason that I like to, or one slide that I like to use to justify why I trust the HHV data as a good proxy for oxygen extraction and the dynamic profile of oxygen extraction is what I have here. So this is our, or these are our HHV data, oxygen extraction data from an exercise transition from 20 watts to the moderate intensity domain. These are the same data, but zooming in and getting into a 60-second frame. And what you can see over here, I think this one, let me see, I have it here, yeah. So this one here and this one here are AVO2 different signals measured in humans and in animals. So basically the same profile for oxygen extraction here and the same profile from oxygen extraction here. And if you look at the microvascular pressure of oxygen and microvascular, intracellular pressure of oxygen and microvascular pressure of oxygen drop with a transition in exercise, you see the opposite responses, which is what you would expect, like a drop in pressure of oxygen. So it seems like this HHV signal is giving us a really good profile of what's happening with oxygen extraction from a dynamic perspective. I won't even get into the absolute values of the HHV signal now because it would take me an extra hour, but we know for certain that we can pretty much trust these dynamic profiles. So before getting into more results, I'll tell you a little bit about this idea that I started describing before, this HHV or VO2 ratio, oxygen extraction for VO2 ratio. So this is the way we estimate that ratio. So basically we have our VO2 data here, breath by breath data in liters per minute, and then we transform that data from 0 to 100% of the response. So it's normalized data, and that has to do with the fact that we are going to also normalize the HHV data. And then we left shift the VO2 data to account for that cardiodynamic phase of the VO2 that I was describing before. And finally, we superimpose the HHV data, the red signal here. So basically, if there is a good matching between those two, the implication is that oxygen delivery is matching the need for oxygen utilization. And I'll show you how it works in our minds here. So basically, when you have oxygen extraction signal that adjusts at a faster pace than the VO2 signal, you get a transient overshoot in the HHV or VO2 ratio, so that overshoot exceeds what you would expect that value to be at steady state, which in this case is 1. And then when you have a perfect matching of those two signals, so the dynamic adjustment of extraction and utilization is the same, then you have no overshoot in that signal. So this overshoot, the implication to us is that there is a poorer provision of oxygen for a given need for oxygen utilization, so that there is a larger reliance on oxygen extraction. So now the next part of my talk would be trying to hopefully convince you a little bit that oxygen delivery to the tissues plays a critical role. And how can we measure that non-invasively? It's by using this HHV and VO2 signal. So this is the first set of data that I'm going to show you. It's part of a training study that we did where we had people, older and young people, coming to our lab to do exercise training. For 12 weeks, we were testing and retesting these people every 3 weeks. I just met a student from, got in touch with a student from Western University back then who was part of my study, so thank you very much. I still owe you for that. We were doing biopsies and some crazy stuff. She never complained, not too much at least. And anyway, so we were measuring VO2 kinetics every 3 weeks, and this is what we found. So we found a very rapid change in oxygen uptake kinetics within 3 weeks of training. So you can see in men, these are the older men and the young men, and same thing, older women and young women. And what you can see is that within the first 3 weeks of training, oxygen VO2 kinetics got much faster, significantly faster, without no further changes thereafter. So both old and young people had the ability to get faster, but everything happened in a very rapid period of time. And then the mechanisms that control that to us were related to oxygen availability, oxygen provision to the tissue. And what you can see here, these are the pre-training data. So you can see that the adjustment of VO2 for a given adjustment of HHV was very different, and there was this gap that resulted in an overshoot in the HHV or VO2 ratio. So less oxygen availability within the tissues to us explained the limitation or the rather slow VO2 kinetics. And what you can see also is that that overshoot tends to disappear with exercise training. And something similar is observed in the older people, but the overshoot is much larger. So it gave us a ratio of 1.25, and then we reduced that ratio with training to about 1.15. So forget about the numbers, they are kind of irrelevant in this context, but the important thing is that people had a faster VO2 kinetics, and what you can see here in these figures is that, so here you have the tau VO2, and here the change in the HHV or VO2 ratio. And they were like perfectly matched almost, both in young and older people. So the faster the VO2 kinetics, the smaller the HHV or VO2 ratio. And we actually plotted that and got this high correlation for both young and old. So explaining this idea, the better you can match oxygen delivery to oxygen utilization, the faster your VO2 kinetics. So one of the many complaints that we were receiving from reviewers was that first, this relationship was driven by data from mean data instead of individual data points, and also the idea that everyone accepted to a given extent that older people could have an oxygen delivery limitation, but young people could not. So young healthy people have to have a fast VO2 kinetics. So we started working on this idea of putting together data from many studies that we had conducted before. And what we found was that we could find people with an average tau VO2 of about 16 seconds. We call that a very fast VO2 kinetics group. Very unusual, there aren't many of those out there. Then we got another group of young healthy people with about 25 seconds VO2 kinetics. And then we got another group of people with about 35 seconds VO2 kinetics. So that's rather slow, even for young healthy people. In the literature, you don't see that often, but we see it, or we used to see it quite often in our lab. And then we even had a group of people with a tau VO2 of 55 seconds. So that's very slow for a young healthy people, 55 seconds VO2 kinetics. According to the previous model, that would make you be one of those diseased populations or an older person. But these were young healthy people with very slow VO2 kinetics. So of course we tested the idea of, okay, let's see what happens with that HHV or VO2 ratio and the overshoot between the two signals. And what you can see here is that good matching of oxygen extraction and oxygen utilization. So the implication is that oxygen delivery is matching the requirements, and then a progressive mismatch between those two signals with progressively slower VO2 kinetics. And then when we look at the ratio, well, obviously the same story. So there was no significant overshoot here, and then a significant overshoot here that got progressively larger when the VO2 kinetics was slower. So again, part of the same story. The faster the VO2 kinetics, the smaller the HHV over VO2 ratio. So better provision of oxygen to the tissues, in our view, had something to do with a faster VO2 kinetics. So that was probably 2010, 2011. I was at the ACSM meeting, and Darren Delory, a previous student working with Don Paterson, had the idea of using this model, the heavy mod intervention. So what he said was, okay, if you think that this ratio is playing a role or is explaining your mechanisms, why don't you try this heavy mod model? So that model implies we do moderate intensity exercise and we calculate VO2 kinetics. Then we do a heavy bout of exercise where we get upregulation of different enzymes related to oxidative phosphorylation and also faster increase in blood flow. And then we measure VO2 kinetics after. And normally, unless the VO2 kinetics is too fast to start with, and this is part of a different story, but when you are in around 20 seconds VO2 kinetics, it's very difficult to make it any faster. But if you are slower than that, then this heavy bout of exercise will likely make it faster. So let's see if that happens, and if that happens, if the HHV or VO2 ratio is reduced. And that's what we did, and we found what we expected to find. There was, in mod 1, a small but significant overshoot in the HHV or VO2 ratio, and the tau VO2 was around 27 seconds. And then in the second mod, the overshoot disappeared, and the tau VO2 kinetics got down to almost 20 seconds. And that's kind of a magical number. That's, in our view, how fast you can be for the most part in terms of VO2 kinetics. And what I'm plotting here is the change in VO2 kinetics and the change in the ratio, and the change in oxygen provision to the tissues. So the more you could improve the oxygen delivery to the tissues, the faster your VO2 kinetics could be. Now, an obvious limitation of that model was that we interpreted that change in the ratio as improved oxygen delivery, but people will still argue, well, you might still be getting a faster VO2 kinetics because of intracellular control mechanisms that are upregulated. So my friend then, well, he's still my friend, but my friend Matt Spencer, when we were doing our PhD, had this great idea of saying, okay, why don't we do that model, but we have the control condition, and then we have an experimental condition when mod 2 is not just mod 2, but it's mod 2 in hypoxia. So we are saying that this, oops, again, I'll get better at some point. We know that this heavy bout of exercise is improving oxygen delivery and also might be influencing intracellular mechanisms. So let's see what happens when we eliminate the oxygen delivery advantage by giving people a hypoxic mixture to breathe. So this is what happened. We had a little bit of an overshoot in the HHV or VO2 ratio with a tau of 26 seconds and 106 for the ratio. And then when we had mod 2, tau VO2 kinetics went down again to this magic number of 20 seconds and a perfect matching with a ratio of 1. When we had mod 2, but in a hypoxic condition, so we got rid of that oxygen delivery advantage. So if it was an intracellular mechanism that was playing a role, so then we should still see faster VO2 kinetics, but that fast VO2 kinetics disappeared. So to us, it was kind of really good, strong evidence that oxygen delivery to the tissues is playing an important role in determining how fast your VO2 kinetics, your oxidative metabolism adjusts to exercise transitions. So the last one that I'm going to show you in relation to moderate intensity exercise is a recent study that one of, I don't want to say my student because she was supervised by Don Paterson and I was acting as a kind of co-supervisor. So she always wants to make clear that you're not my supervisor. So sorry, Kate. So what Kate was doing was this idea, okay, let's look at the rapid adjustment or changes in the VO2 kinetics response and what's explaining those changes. And the reason for doing that is because there is animal data supporting the idea that you can get a much faster, a greater vascular responsiveness 24 hours after an acute bout of exercise or short periods of training, and that response can be upregulated for 48 to 72 hours. So what we did, we had people coming to the lab and we measured, this is young and older people, we measured VO2 max, then we measured VO2 kinetics, and we also calculated this ratio, HHV or VO2 ratio, and also FMD, flow-mediated dilation response, to have a measure of what's happening with the vessels in terms of vascular reactivity. So then we trained people within a week, 3 training sessions, 70% of VO2 max, young and old people, and then we retested them 24 hours after the exercise intervention, 48, 72, and 120 hours after. So we wanted to see how quickly this change could happen and for how long it would be sustained, and potentially what were the mechanisms controlling that change in the response. So what we saw, and I apologize, I know it's a messy slide. So these are the older people here, and the young are below. So VO2 kinetics got significantly faster 24 hours after, remained faster 48 hours after, about 3 exercise training sessions, and tended to be faster, but it's a p-value of 0.07 after 72 hours. And you can see that the older people seem to be coming back, but the young people still seem to be responding well to that intervention. We measure our HHV or VO2 ratio, and we show the same idea. So a smaller ratio after 24 hours, sustained for 48, and still significantly different 72 hours after. But you can see the same trend, like older people, the ratio is coming back. And the other measure that we did, the FMD, so you can see that 24 hours after, there was a better or larger FMD response. So let's call it better vascular responsiveness, at least in these conduit arteries. And that response was sustained for 72 hours as well. But you can see that that response is disappearing. So again, our interpretation is that oxygen provision to the tissues is important, and probably vascular responsiveness is mediating that improved oxygen delivery. But again, all this data put together, basically what gave us was like nice ways of explaining the mechanisms controlling VO2 kinetics by using simply VO2 data and oxygen extraction data from a NIRS device. So now I'm going to try to talk a little bit, and this is part of an introduction to our next speaker, Daniel Keir. So I'm going to be talking about ramp incremental exercise. So what's happening with oxygen extraction, oxygen utilization, and what are the implications for oxygen delivery from this data. So when we are talking about central or more systemic responses, we have a linear relationship that is very well established between VO2 data here and cardiac output. So they increase kind of linearly, and these are data from another study that we did. So this linear increase in VO2 for a given, in cardiac output for a given increase in VO2 implies a hyperbolic response for a VO2 difference. So these are the profiles that are typically described when you are looking at systemic data for VO2 and cardiac output, and then a VO2 difference. So about 10 years ago, people working with Tom Barstow's group and David Poole's group started looking at the oxygen extraction data, the HHV data from NIRS, to try to interpret what was happening with oxygen delivery to the tissues from a muscle perspective and not from a systemic perspective. And what they found was that oxygen extraction described a completely different profile as to what we see systemically. So there was a rather linear increase in extraction, and then it tended to plateau, and that implied that the oxygen delivery could be or resemble an inverse sigmoid profile. So that linear increase in VO2, in cardiac output that we see systemically doesn't need to be true within the muscles. And this is within the vastus lateralis muscle. Things might change depending on the muscle that you use, but again, that's part of what Dan will be discussing soon. So what we wanted to do, I always say we didn't invent anything when we came up with the next study that I'm going to present, but something kind of neat that we did. So these people had shown oxygen extraction. They call it a VO2 difference here based on a calculation that they did, but basically these are oxygen extraction data. So we proceeded with a study where we measured cardiac output minute by minute and then estimated the VO2 difference. So this is what we called our systemic responses. And then we looked at the peripheral responses. And not surprisingly, these two are the same. So that inverse sigmoid response adjustment in blood flow within the muscle, that is not exactly what you typically see when you are talking about systemic exercise. But another very important finding was looking at this breaking point here. And some subsequent studies, like also people working with Tom Barstow, Jan Boone, he works in Belgium now. He started modeling that response, that HHV response during ramp incremental exercise as a sigmoidal response. And we were looking at the signal, and we were feeling that that wasn't the best possible fit. So we came up with a different strategy and played around with the data. And what we established in this paper was that the sigmoidal response, and sorry, these two data sets are basically the same. One is plotted as a percent of VO2, the other one as a percent of, of power output. But basically what we showed was that the sigmoid fit not only left a lot of data points untouched in that fit, but also overestimated and underestimated the baseline and the steady state response. So we decided that a better fit could be considering this as a double linear component. And the double linear component was a much better fit and left us with this breaking point here. And we were curious about what that breaking point represented. And in a study that we published where we were comparing oxygen extraction in men and women, we added a piece of information that was interesting to us, showing that the VO2 associated with that breaking point was the same as the VO2 associated with the respiratory compensation point in both men and women. And simultaneously, Silvia Pogliagh had done a much better job collecting data in over 100 people, where she identified with her group that that correspondence between the breaking point and the RCP was almost perfect. So there was a kind of clear evidence that these 2 points coincided. And I'm going beyond my time, so I'm going to try to rush it now. So one of the things that we were wondering was, okay, what is that breaking point happening? It seems to indicate respiratory compensation point or to be close to that point. And in our head, we said, okay, respiratory compensation point implies a lot of things happening from a muscle metabolism perspective that will increase blood flow within that specific region. So that increase in muscle metabolism would imply an increase in blood flow. So maybe local blood flow is increasing, and that implies that then we see a plateau in this signal. So now we are experimenting with this model where we have people coming to the lab. So these are the oxygen extraction signal. We have people sitting on the bike at baseline, and then we occlude the femoral artery, so no blood flow to the artery, and in response to that, we see a massive increase in oxygen extraction. Once that signal plateaus, we leave it for about 5 minutes, then we release, so oxygen extraction is reduced. At some point, it stabilizes, and once we have that data steady and stable, we start a ramp incremental test to exhaustion. And in response to that ramp incremental test, we get the same response, like a plateau, and then in the HHV signal, and then we have another occlusion. So this is an occlusion, femoral artery occlusion right after the end of the test. It doesn't feel that good. Yeah, we aim for 2 minutes. Everyone who is in my lab or nearby will know that I started screaming like a little boy after 45 seconds. So before Kalain starts telling you that, I'll say it here up front. Yes, I'm weak. So yeah, joke is over, Kalain. So we were saying, okay, if there is an increase in blood flow, so there has to be potential for further oxygen extraction, so there might be an oxygen extraction reserve. And I'll show you this last slide here, where I show you the model. We have occlusion there. We have the increase in oxygen extraction here. Then this is our ramp incremental test. The HHV signal plateaus. Then we occlude again, and this is what we get. Like the oxygen extraction, even before the ramp incremental exercise, exceeds the plateau. And then after ramp incremental exercise, there is a further increase in oxygen extraction. So there is a reserve for oxygen extraction, which implies that to us, blood flow in that region has to be increased. So again, further emphasis on this idea, looking at systemic versus peripheral components of these adaptations is important. And I'm going to finish with my concluding remarks. Hey, we can do a pretty good job at learning something about oxygen distribution, oxygen provision, oxygen extraction by using some relatively inexpensive and friendly to use non-invasive tools. So we are very excited about the work that we have been doing for the past 10 years. We keep on developing new ideas. But yeah, it's fantastic to me that with, again, relatively simple tools, we can do a really good job. Before I finish, I'd like to do some acknowledgement. I have to thank Don all the time that I present, even if he's not here. He's been the greatest person to work with and hang out with as well. Also, participants in our studies, like we are nothing without people putting their time and making a big effort so that we can collect some neat data. So thanks to them, even, well, at least there is one of them that is here. And then I put a very broad graduate students and colleagues. Working with Silvia, with Dan, with many people has been fantastic. And I always thought that people were overstating stuff when they were thanking students, graduate students. And now that I'm here and I'm not doing much of data collection, I appreciate more and more what they do. Fantastic group of people working with me, so I'm very thankful. So funding sources, universities, and especially for this symposium, I'd like to acknowledge COSMED. Not only they give me a really good tool to measure VO2, but they contributed to this symposium so that we could bring Silvia for the presentation. So yeah, that's the acknowledgement. Thank you very much.
I think since Juan was exactly on time, we won't stop for questions in between. I can tell you that everything he said was absolutely true. Now that I'm retired and living in Canmore, I go hiking or snowshoeing once a week with the seniors. That's all fine, and then my kids come out and they want to go hiking with me, and in the first 5 minutes, they're 400 or 500 meters ahead of me. Their kinetics are 24 seconds, mine are 40 seconds. So it's heavy exercise for me for the first 5 minutes of the hike. And that provides me a warm-up. I stop for a little bit. Then when I go again, my kinetics are faster and I can keep up to them. But Dan Keir will now come forward, and in his talk will tell you from different exercise intensity thresholds why I can't keep up to them when we get to the top of the mountain. Daniel.
What an honor it is to be introduced by Dr. Don Paterson. Since coming to Western and him being a mentor of mine, I've tried to emulate my my scientific career in his visage, and while I'm not even coming anywhere close to his research abilities, I was able to at least copy his hairstyle. So at least I did that, Don. So for today, I'm going to talk about using VO2 and NIRS to stratify exercise intensity. My outline will be to review the traditional criteria for aerobic exercise intensity prescription. I'm going to present a case study of pulmonary oxygen uptake kinetics to highlight the shortcomings of the traditional approach, emphasize the need for threshold-based exercise intensity prescription. I'm going to discuss the use of NIRS and VO2 for threshold-based exercise intensity stratification, and finish off by recommending strategies to optimize prescription for aerobic exercise training based on these VO2 and NIRS signals. So these are the guidelines. You open a standard exercise physiology textbook. These are the guidelines by which we currently stratify exercise intensity. So this operates this, We're operating under the assumption that from resting VO2 to VO2 max during an incremental exercise test, that we can that this is a linear response. And so if we normalize that to 100, from 0 to 100%, and prescribe an exercise intensity based on a percentage, then for different individuals, we can have them exercise at the same relative intensity, and then the implication or the assumption is that they'll be exercising at the same, the metabolic and physiological stimulus will be the same at that intensity. And so we can stratify exercise intensity into these categories. And by choosing a percentage of VO2, we can have those two individuals exercise at the same intensity. So an underlying assumption of this is that VO2 increases in a linearly with power output. And I would like to say that all of the data that I'm going to be presenting is in terms of cycling. They will be cycling data, but if your interest is in running or swimming or any other form of locomotion, think about, you can substitute power output for a different form of intensity if you want to think about it in terms of speed. It will apply the same across those different modalities. So this is the standard assumption. So here's the shortcoming of this exercise intensity range-based approach. So I'll provide a little example here. We have two individuals who are asked to cycle for 30 minutes at 75% VO2 max. So we have our VO2 reserve. So they've both performed an incremental exercise test. We've looked at the linear response. We've figured out a VO2 associated with 75%, and we can prescribe a power output based on that VO2. So here we have subject. Here we have subject number 1, his VO2 max is relative of 50. Subject number 2, again, relative of 50, so they're relatively the same fitness level. We have them exercise at 75% VO2 max. And let's see what we have here. So we look at the first individual, and this blue line represents the VO2 that we expect for the power output that we prescribed. So we expected that a power output of 280 would give us a VO2 of 3.4 liters per minute. So you can see in this individual that the VO2 increases in an exponential-like manner, but the VO2 steady state projects above where we would expect based on that linear power output relationship. But the person was capable of exercising for 30 minutes at that power output. Now again, we've prescribed 75% of VO2 max for the second subject. However, you can see that VO2 not only shoots above where we would expect based on the power output that we chose, but it also continues to increase until VO2 max, 4 liters per minute, is achieved. And obviously there's implications for exercise tolerance. This individual is not even capable of performing 75% of VO2 max for 30 minutes. So although we think that both of these individuals are exercising at the same relative intensity, clearly the metabolic and physiological consequences of exercising at that intensity are not the same. So I'm going to now speak to subject number 2 here and just give you some data collected on that particular individual to illustrate further the shortcomings of this relative intensity approach. So these are exercise transitions to different power outputs within this subject. And again, this is constant power output exercise performed for 10 minutes. So what I'd like you to note is that as the power output increases, VO2 increases, as we would expect. You need more oxygen to resynthesize the ATP requirements, but the VO2 profile begins to change. So these might look similar, but this one, you can tell, takes a longer time to reach a steady state, and at these higher intensities, you don't even reach a steady state at all. And there are obviously implications for exercise tolerance at the higher power outputs. So this is an example of how exercise intensity is really, oxygen uptake is not a linear function of power output or exercise intensity. So for the untrained eye, you might not have seen the subtle differences as as that person performed higher power output. So what I've done is I've extracted 3 of those profiles from that individual, and I'm going to compare them to the VO2 response to incremental exercise. So we can see at the 40% VO2 max, we've prescribed a work rate based on that 40%. And then when we do a transition to the power output associated with 40% VO2 max, we can see that the VO2 achieves a steady state as we would predict from the incremental exercise. So this appears to be a linear response. However, when we exercise at a higher intensity, what we see is that the power output that we chose to elicit a given VO2, now at that power output, VO2 exceeds what we would expect based on that linear relationship. And also, the VO2 response takes longer to reach a steady state. It does reach a steady state, but it takes longer to do so. And this is referred to as the VO2 slow component. So this extra component, more VO2 is required for a given power output. If we now go even further and select a power output corresponding to 80% VO2 max in this individual, we can see that the VO2 response projects much further. The slow component is larger at that higher intensity. And again, there are implications for exercise tolerance. In this case, the individual on the x-axis, I think it's about 8 minutes of exercise, was not able to complete 8 minutes of exercise at that intensity. So this highlights the issue of transferring data, the VO2 power output response from incremental exercise into constant power output exercise, which is often what we're prescribing based on incremental VO2. So when you plot the end exercise VO2 of all of those curves and superimpose them over the incremental exercise test, so here are the incremental exercise test breath by breath VO2 data for that individual, what you see is that for the lower intensities, the VO2 is a relatively linear response as a function of power output. And I would like you to note that those data points are shifted to the left, and that's because the ramp incremental test does have a kinetic component, which you have to factor in when you're assigning a specific power output to a specific VO2. And what we see as we increase the intensity, the VO2 falls further and further from that line in the constant power output paradigm, and a slow component emerges, and the VO2 slow component gets bigger as the greater the exercise intensity. And at some intensity, it doesn't matter what power output we choose, eventually VO2 is going to increase inexorably until you reach VO2 max. So clearly, VO2 is not a linear function of power output, and there are implications for exercising at different intensities within a given individual, and it's not just cannot be simply stratified in terms of a relative or percentage VO2 max. So what is the underlying physiology? What's changing here? Why is VO2 reaching a steady state that we expect, and then all of a sudden we start to see this VO2 slow component? What's happening? What is the underlying physiology that's occurring during an incremental exercise test? So when we do measure VO2 during incremental exercise, we're lucky enough to also gather other gas exchange variables. So if you're to look at the data within the lower regions, you see that VO2 increases, VCO2 increases relative to VO2, ventilation increases to meet the oxygen demands. And if you were to measure arterial lactate concentration, you wouldn't see any change in arterial lactate. And these are nice linear responses. However, at some point, at some VO2, the glycolytic pyruvate production exceeds its rate of use in the mitochondria. And what happens is you have a net increase in lactate within the muscle, and that's rapidly transported out into the blood. So lactate, of course, is associated with a hydrogen ion increase because it's completely dissociated at arterial pH. So we get an increase, anytime we get an increase in lactate within the blood, we get an increase in hydrogen ion. And of course, we have a buffering system in place to eliminate that increase in hydrogen ion and maintain arterial pH constant. So what happens at that point now is we force this reaction, the carbonic anhydrase reaction here to the left, getting an increase in CO2. And we can measure that change in CO2 relative to VO2 now that we're producing non-metabolic CO2. And we can call that, or it's traditionally called in the literature, depending on which graph you look at and how you discern these breakpoint changes, the gas exchange threshold, the ventilatory threshold, the lactate threshold, it's been termed the anaerobic threshold. But we can assign a specific VO2 to that threshold where clearly the physiology is changing above versus below that threshold. And because the VO2 response is linear, VO2 work rate response is linear in that lower range, we can quantify precisely a power output that will elicit the lactate threshold, so the VO2 associated with the lactate threshold. So now if we go back to our constant load exercise paradigm and we superimpose where our detected lactate threshold is, and we compare the VO2 responses to the blood lactate responses, and you can see the line here, that's 10 minutes, so this goes to 10 minutes, but this is extended to 30 minutes of exercise. You can see that for all exercise intensities within the moderate intensity domain, there is no net lactate accumulation. So the metabolic stimulus appears to be the same within that exercise intensity domain. The implications for VO2 are that VO2 increases in an exponential-like manner towards a steady state, and that steady state is predictable based on the VO2 work rate relationship from incremental exercise. If we transition now to a power output above this lactate threshold, what we see is now we get an elevated, so these data points correspond to 180 watts. There's net lactate accumulation in the blood, but we're able to maintain that in a steady state. So there's a perturbation here, but we're able to maintain homeostasis and exercise at that intensity for a prolonged period of time. And so we can define a set of power outputs, a range of power outputs that have similar physiological and metabolic consequences, and these are those that are below the lactate threshold. So we refer to these as moderate intensity exercise. So the VO2 reaches a steady state, there's no net lactate accumulation. The implication for exercise performance is you can exercise for hours, and the VO2 power output relationship is linear. So you can use incremental exercise. You can assign a specific power output to elicit a specific VO2. So if we go back to our incremental exercise and we go a little bit further, we push this person a little bit further, increase the power output, we increase the VO2. We see that at some point, lactate continues to increase, and we're able to defend arterial pH. But what happens is, at some point, the bicarbonate buffering system is no longer capable of buffering that response. The system is not capable of maintaining homeostasis or buffering the metabolic disturbance. And so what happens is we continue to get an increase in lactate. We're no longer able to compensate for this arterial acidosis. Bicarbonate is reduced, and the only way we can buffer or partially attempt to buffer this response is by eliminating CO2 and driving the reaction to the left by the process of, or the principle of mass action. And so what you see is an increase, you may see an increase in CO2 again relative to VO2, but the major thing that you might see is you see this extra increase in ventilation, which has been termed the second ventilatory threshold. This point has also been called the respiratory compensation point because there's partial compensation for the metabolic acidosis. And so regardless of how we measure it, we can take the second ventilatory threshold, the respiratory compensation point, but we can find a VO2 that's associated with this important threshold. The problem now becomes when we want to translate that VO2 from incremental exercise and figure out a power output that will elicit that particular metabolic intensity, we're unable to do that because the VO2 work rate relationship is not linear. So if I wanted to find a VO2 associated with 3.4 and I went to my graph here and went down, you see if I chose that power output, I'd actually end up with a power output that exceeds the respiratory compensation point. And again, that's going to have a different metabolic, that's going to be a different metabolic and physiological stimulus. So now we can superimpose this second threshold onto our constant power output graph, so a VO2 associated with 3.4. And when we look at the lactate responses, we can see that for the exercise, for all power outputs between this metabolic rate and the lactate threshold, we get an increase in a net accumulation of blood lactate. However, we're able to maintain that at a stable level. So metabolism, though the stimulus is greater, we're able to maintain that exercise intensity for a prolonged duration. If we exercise above this respiratory compensation point, what we see now is that the lactate increases. It doesn't reach a steady state, and obviously there are implications for exercise tolerance. We're not able to sustain the exercise above this critical metabolic intensity. And so now we can define, again, a range of power outputs, which we call the heavy intensity domain, where the VO2 does reach a steady state. Lactate, there is net lactate accumulation. There's lactate accumulation, elevation, but it does reach a steady state as well. The implications for exercise tolerance within this domain is you can exercise for a prolonged duration. It doesn't feel as good, but you're capable of exercising for a prolonged duration, and I have 30 plus minutes there, just as a, most people don't like to tolerate exercise for too much longer in that domain because it doesn't feel great. There is a significant metabolic stimulus. But we can't predict a power output associated with that VO2 because of the slow component during incremental, that's not evident in incremental exercise. So assigning a power output to elicit that VO2 is problematic. And finally, we can define, again, another range of power outputs that we describe as severe intensity exercise. And the implications of exercising within this domain are VO2 doesn't reach a steady state. Exercise tolerance, exercise exhaustion ends with the achievement of VO2 max. There is no lactate steady state. Lactate continues to increase until exercise is no longer tolerable. The duration is limited, so you could only potentially, depending on how far you are away from that critical metabolic threshold, the further away you are, the less time you can exercise within that domain. And again, it's really difficult to predict a power output that from the incremental exercise within that domain. So I would like to say that this schema of exercise intensity has been around since probably the late 1980s. However, we continue to ignore it and continue to prescribe exercise in terms of percentage VO2 max in relative terms, when really it's inappropriate because we can't really predict, especially for these higher exercise intensities, we can't predict a power output that will elicit a specific VO2. And so now for this individual, we've now defined the entire spectrum of exercise intensity from rest to VO2 max. Not as a percentage, not as a relative percentage of VO2 max, but in terms of these exercise intensity domains. So clusters of power output that give you the same physiological and metabolic stimulus. So for the most part, because the VO2 work rate relationship is linear in the moderate domain, we can measure the lactate threshold from our gas exchange data by measuring lactate, and we can define a specific power output that's associated with that particular critical metabolic intensity. The issue now becomes when the relationship is no longer linear, how do we define this heavy severe intensity boundary? And power output is one of the issues. The other issue is what exercise indice, what threshold best describes or is best representative of this critical heavy severe boundary. So I'm going to focus the rest of my talk on how we might identify a power output and what indice is best reflective of this critical exercise intensity. So just to give you a little bit of background, so defining the heavy severe boundary, I'm going to refer to it as a critical intensity concept because really the implications are that you're not able to sustain exercise above this intensity, but you are able to sustain exercise for prolonged periods of time below this intensity. So the physiological definition is the highest metabolic rate that can be sustained entirely by oxidative metabolism and the highest intensity at which arterial pH can be defended, so you can maintain homeostasis. So in implications from a training perspective, the critical intensity demarcates sustainable exercise from unsustainable exercise. So clear implications for exercise tolerance above versus below this intensity. The higher the task-specific intensity is from the critical intensity, the sooner exhaustion will ensue. So it's almost it's like a hyperbolic type relationship. So the further you are away from your critical intensity, the further the power output is from that the power output associated with that critical intensity, the less time, the shorter the duration you'll be able to exercise for. The higher one's critical intensity, the more aerobic work can be done for a given distance. So if we had two runners, one's critical intensity corresponded to a speed of 15 kilometers an hour, another to 12 kilometers an hour, and we asked them both to finish a 10-kilometer race, the one with 15 kilometers an hour could do that solely using oxidative sources in 40 minutes, whereas the other would do it in 50 minutes. So clearly it's an important index of aerobic fitness. So this is the real challenging question in the literature is how can one identify the critical intensity? So anybody who's studied classical exercise physiology and has opened a textbook would probably be familiar with a lot of these different concepts. And all of them have been proposed to reflect in some way this critical intensity. And I'd like to say that as many studies have compared different thresholds, but as many studies as there are that have said that they're similar, you could probably find as many that say that they're different. So it's a real challenge to reconcile the relationship between all these candidate thresholds to identify this critical intensity. And I'm going to spend a little bit of time talking about our contribution to this body of literature through this NIRS-derived threshold, the HHB breakpoint. So just to remind you, this is the data that I'm going to be presenting are measured during cycling on the vastus lateralis muscle. And so we think about HHB in terms of oxygen extraction. So if we take the Fick equation and we replace VO2 difference with HHB, then the relationship between VO2 to Q represents our HHB. So essentially it's our oxygen extraction. And our interest in this area kind of happened serendipitously because we were really just interested in characterizing this response. Juan Murias talked about this in his previous in the presentation you just saw. So the traditional way to model this was with a sigmoid, and Matt Spencer came up with the idea of saying, well, why don't we try and quantify this with a double linear, see which one fits the data better. Now, I'm not going to stand up here and say that this model is perfect. All models have inherent assumptions and limitations. But what the advantage of this double linear response is you can characterize the point at which the rate of increase is reduced. And it's a pretty reproducible phenomenon that we see. So after this paper, we thought, well, what does this threshold represent? What does it mean? What are the underlying mechanisms? And how might we be able to adapt this for exercise intensity prescription? So the first paper where we compared this to the respiratory compensation point, because we noticed that it occurred roughly around 75 to 80% VO2 max. So our hypothesis was that perhaps the VO2s associated with this respiratory compensation point would correspond with the HHB breakpoint. And an important aspect of this study is that we did it in terms of VO2 instead of power output, because again, at those higher relative intensities, the VO2 work rate relationship is not linear, so it's difficult to assign a particular power output to a particular VO2. So we noted reasonable limits of agreement. You can see that the mean bias was not different, so our conclusion was that they appear to be occurring at the same metabolic rate. Dr. Pogliagh did some good research looking at, okay, in 100 subjects, how close does the respiratory compensation point match with the HHB breakpoint, again, in terms of VO2, and you can see that there's a nice relationship there, equal distribution of individuals on the lower side and the upper side of the line of identity. So we're pretty confident that the underlying physiology associated or the underlying mechanisms associated with the two breakpoints were related in some way. She also went on to compare it beyond the respiratory compensation point to another marker of aerobic metabolism, which is the maximal lactate steady state. And when she found the VO2 associated with both, again, she saw a nice relationship between the two and equal distribution, signifying again that they reflect the same metabolic rate and may share common underlying physiological features. So for the mechanisms, if you think about the HHB breakpoint simply, and it's related to VO2 to Q, if we have a linear increase in peripheral blood flow, and again, when we're thinking about the NIRS breakpoint, we're measuring something that's very local. It's a local response because we're measuring it directly above the muscle. So if local blood flow increases linearly and VO2 increases linearly, at the systemic level for AVO2 difference, we get this type of relationship. Well, when we compare that to the HHB breakpoint or the HHB profile, it doesn't really look all that similar. So we weren't really in favor of this hypothesis. Another hypothesis is that we had was that at that point, blood flow is exceeding the rate of increase in VO2, so a sort of relationship like this, which would give you a response that looks similar to that. to the one we observed. A third hypothesis that we had was that perhaps blood flow is increasing linearly, but VO2 curtails towards that, beyond that, that HHB breakpoint, possibly because of a recruitment of type 2 fibers that are less oxidative. Again, we didn't really like that hypothesis, so we focused some subsequent research on testing whether this hypothesis are true or not, and Juan presented some of those data earlier. So I'm not going to get into how we derived this curve, but basically what we did was the relationship that we think we're seeing is that we're seeing a relative hyperemia beyond this HHB, or at this HHB breakpoint. And I'm just going to speak to why it might be related to the respiratory compensation point, because that was really my interest, because I'm interested in integrative physiology. So the respiratory compensation point coincides with this reduction in arterial CO2 when we hyperventilate. So we thought that maybe CO2 was a mechanism by which we're getting an increase in blood flow. So if we reduce a vasodilatory stimulus in the body, perhaps we're getting a redistribution of blood flow to the muscle. We're yet to completely test or to test this hypothesis, but there are some encouraging data that came out of Boone's lab where they were looking at cerebral deoxygenation, and coincident with that respiratory compensation point, you see a very sharp rise in oxygen extraction. So as we know, or you may be aware, CO2 is a potent vasodilatory stimulus in cerebral circulation. So if we remove that dilatory stimulus, we reduce blood flow, and we get an increase in extraction. So if that's happening in all the other non-metabolically active tissues, this reduction in CO2 could be related could be a mechanism by which we get an improvement in local muscle oxygen delivery. So really what this says is that if you think about it, it's really an integrative system. So that made us think about, okay, well, how do all of these thresholds fit together? No one had really done a comprehensive study of looking at all of the measuring the VO2s associated with all of the thresholds and comparing them. So we hypothesized in this study that we published in MSSE that the VO2 associated with the HHB breakpoint would equal the VO2 associated with RCP, MLSS, and critical power. And no study had looked at all of these and measured the metabolic rate associated with all of these at the same time. So what we did is we had individuals come into the lab. They performed 5 exhaustive exercise trials at a constant power output, from which we derived the time power output relationship. Of course, the asymptote of this represents the critical power. We had individuals exercise at that critical power, measured VO2 and lactate. If the lactate did not reach a steady state, we dropped the power output by 10 watts until we found the power output that was associated with a lactate steady state. And so we could also find the VO2 associated with maximal lactate steady state from this. If the critical power did reach a steady state, the lactate did reach a steady state at the ride at critical power. We then increased the intensity or the power output by 10 watts in a subsequent ride to determine the maximal lactate steady state. So from this, we could determine the VO2 associated with critical power and maximal lactate steady state. We also performed incremental exercise, and we could determine the respiratory compensation point from that, and also measured the NIRS HHB breakpoint and determined the VO2 associated with that. So this is just a representative subjects data. So a lot of testing sessions, but we were able to come up with both a VO2 and power output associated with all of these different indices of exercise intensity. And here are the group mean data. And what you can see for our estimation of critical power, we found that in 11 subjects, 5 of them, the critical power did represent the maximal lactate steady state. In 3 of them, it was the estimate was higher, and in 3 of them, the estimate was lower. So when we looked at the VO2 associated with maximal lactate steady state and critical power, there was no difference in the VO2 associated with those parameters. And when we also looked at the respiratory compensation point and the HHB break point, again, we found that they all tended to occur around the same metabolic rate, suggesting that the underlying physiological mechanisms or underpinnings associated with these thresholds are common. Interestingly, when we, I didn't include it there, but when we did look at power output, these were different, which doesn't surprise me because, again, the HHB break point and the respiratory compensation point are determined from incremental exercise, and of course, it's almost impossible to determine a power output associated with the VO2 that you've determined for the respiratory compensation point or the HHB break point because of the action of the VO2 slow component ruining the linear relationship between VO2 and power output. So this is sort of our current hypothesis or a conclusion from that study, in that all of those different thresholds reflect a specific metabolic intensity, but are different manifestations of the integrated system. So what might happen is oxidative metabolism can no longer satisfy the ATP resynthesis requirements of dynamic muscle effort. That may be related to inadequate glycogen, inadequate substrate delivery to the mitochondria, or ATP delivery to the contractile units. As a result, there's an accumulation of metabolites, inorganic phosphate, ADP, lactate, those associated with mechanisms by which we fatigue or by which there may be contractile impairment. There's transport of lactate out of the muscle, and that exceeds its clearance from the blood, so we get a net lactate accumulation. And then, of course, there's an overload of the body's compensatory mechanisms to sustain or to buffer that metabolic acidosis. And so local metabolic changes may induce these systemic effects that we're seeing. So when you really think about the physiology and then you think about these different candidate indices, you can see that they do actually fit together quite nicely. So one of the issues then becomes, okay, well, if any of these we can use any of these different measures to find the VO2 associated with this critical intensity, what is the best way to define a power output associated with that intensity? All of the methods have inherent inaccuracies. So a study that we just published in APNM out of Dr. Murias' lab, we looked at actually measuring this maximal metabolic steady state. So we looked at VO2 and lactate, and we tried to find the power output associated with the highest steady state in both of those signals. And so when we found that power output and we compared it to critical power derived using a hyperbolic power time relationship, what we found was that the power output derived from critical power tended to exceed the power output associated with that maximal metabolic steady state. And the mean bias was about 20 watts difference. So it's quite a bit. So what this shows is that people often think that critical power may be the best way to define this critical intensity, but in this study anyway, we saw that there was a bias associated with defining this metabolic intensity from the critical power test. So we subsequently had the hypothesis that perhaps we don't need all of these tests to measure critical power. What if we just ask the individual, well, why don't you tell me what the highest power output you think you can sustain for 30 minutes is? And when we did that, we actually found a very interesting relationship when we compared it to the maximal metabolic steady state, the power output associated with the maximal metabolic steady state. So we found that the mean bias was not different. We saw that many people were actually capable of selecting that particular intensity. Obviously, there were some that were further from others, but suggests that this may be an accurate way of doing it, and it's definitely a lot less cumbersome, and it requires a lot less visits, so this might be a promising way by which to identify or assign a power output associated with that metabolic intensity. But really what it tells you, what it all boils down to is, regardless of what test you use to identify that critical intensity, if you don't do a validation trial and measure lactate or measure VO2 and actually identify that is the highest power output that will elicit a steady state in these in the physiological and metabolic profiles, then you really don't know what you're getting into. So it's difficult to actually prescribe an exercise power output around this intensity. But what I would recommend is if you are interested in having this as using this in an experiment or using this for training purposes, that you actually validate that, you do a validation trial to make sure that that intensity reflects what you want it to reflect. So implications for health and performance, just to summarize, we can use exercise testing, we can use VO2 and NIRS measures to determine this critical intensity through the HHB breakpoint and respiratory compensation point. So we can perform our incremental exercise tests, we can determine the RCP and the HHB breakpoint, and we can use that VO2 as a way to stratify exercise intensity. And so we can compare the VO2 associated with those responses pre versus post training to see if our training is effective at increasing aerobic capacity in addition to VO2 max or whatever other parameters you're interested in. And then we can reevaluate to check training effectiveness. In terms of exercise prescription, you can only really estimate this intensity using the different tests. When you're looking at assigning power output from incremental exercise to elicit a specific VO2, you have to consider the kinetics and the slow component when aligning VO2 with your measure of exercise intensity. And the only way really of prescribing, figuring out where this intensity lies is by doing a validation or verification trial while you measure lactate, VO2, and potentially there could be a role of looking at RPE as well. And then you can use this intensity as a benchmark for exercise training. So I just kind of propose here a little bit of a paradigm. Because exercise is tolerable below this exercise intensity, if you're doing continuous type training, you want to, if the subject or the athlete or whomever you're training is willing, you want to try and get as close as you can to this critical intensity, because you can exercise for 30 minutes or longer, depending on the motivation of the individual. So you're going to get the greatest metabolic perturbation, which should result in adaptations. So in terms of looking at interval training, if you go slightly above this threshold, you're able to sustain this for longer periods of time. You may have the individual exercising at VO2 max for longer as a result of working within this severe intensity domain. And you could look at long-term or long-distance intervals in that sense. And then, of course, the higher you are, there's this hyperbolic relationship, and so the higher the intensity above the critical intensity, the shorter the duration. So you can integrate this concept into traditional exercise training. So to summarize, the assumption that VO2 increases as a linear function of exercise intensity is incorrect. Should really abandon the use of prescribing exercise in terms of percentage VO2 max. The relationship is not linear, and you cannot predict above the lactate threshold. The relationship is not linear, and you cannot predict a power output that will elicit a given VO2. So again, exercise intensity, I can't stress it enough, should not be prescribed in terms of percentage VO2 max. It should be prescribed relative to these exercise intensity domains. Gas exchange, VO2, NIRS derived data from incremental exercise can be used to identify where these domains are in terms of VO2. We can prescribe moderate exercise intensity because there's no slow component below the lactate threshold based on incremental exercise. But to determine the heavy intensity, severe intensity border, we can only determine that by performing one of these other tests, coming up with a best guess as to where that power output is, and then performing a validation trial to make sure that the physiology and metabolism it is the power output associated with the highest physiological and metabolic steady state. So the last thing I'll finish off with are just a few tips for NIRS. I don't think, if anybody's interested in now adopting the NIRS into their lab and has not used it, the target muscle should be the prime mover. So we use vastus lateralis for cycling. So if you're interested in running, use something else. And I'll just finish there. I've gone way too long. So thank you very much for your attention.
So Dr. Keir has explained that my kids are very nice to me when we're hiking up those mountains near Canmore. They go at a moderate intensity. I go at a heavy intensity to try to keep up to them, but I've been able to self-select that intensity so I don't go into the severe domain. It would be unsustainable and I wouldn't get to the top of the mountain. Our next speaker, I don't think we warned her when we asked her to come that she not only had to fly across the ocean, but across an entire continent as well. Dr. Silvia Pogliagh has come from the University of Verona in Italy, and she'll be changing the direction a little bit and talking about VO2 as a vital sign related to health, and should practitioners use this as a vital sign. Dr. Pogliagh.
Thank you very much for making it happen. So we have to switch from the lab to a physician cabinet now. So it's a totally different point of view. So you'll hear about why, if and why, practitioners should consider using VO2 max as a vital sign. So first of all, what are vital signs? They're basic measures, parameters, that are indicative of the current functioning of the body. Doctors will be measuring these parameters, and the main ones are blood pressure, pulse, respiration, and temperature. Your doctor will be measuring those parameters upon your first visit and upon checkup. The doctor will also be interested in secondary vital signs, which can range from sleep to sexual activity to alvus, menses, a number of different functions. He also might be interested in asking about pertinent lifestyle choices, such as alcohol use or smoke. And this will provide your physician a complete, rather synthetic, but realistic picture of your current health and on your prospective health as well. So, okay, this maybe is interesting, but why is it pertinent to our interest today? You know very well Exercise Medicine Initiative. Embedded in this initiative, ACSM is asking physicians to become more active in improving their patients' lifestyle in terms of the amount of physical activity that they perform. In order to do this, physicians should be able to prescribe exercise quantitatively whenever the person is ready for that, meaning they're in the contemplative or active phase. But in reality, considering the prevalence of inactivity in our societies, oftentimes you need to take a step back and work on the awareness of individuals regarding physical activity importance. So in order to promote awareness, physicians are encouraged to speak to their patients about the importance of physical activity, often using a parallel with food needs. You know that there are dietary reference intakes. Maybe we should be talking about physical activity reference intakes. Patients should also be made aware of the fact that inactivity is harmful, and each one of us should be getting adequate amount of physical activity vitamin. So one of the strategies to improve awareness of patients is asking the right questions. So ask about physical activity. The reason for specifically targeting this item is that it's easy to monitor. There are standardized questionnaires by which physicians can record physical activity habits. It's relatively time efficient. Also, it's universally known as a valid indicator of relative risk of all-cause mortality. And another important issue is that physical activity patterns can be easily translated into a prescription. So if you have a physical activity volume to relative mortality relationship, you can then prescribe the right amount of exercise that will reduce your risk of dying for any cause. But is this the right thing to do? Should we just be asking people if they're exercising enough? We definitely would not do that in the case of other diseases. Nobody's dreaming of monitoring the risk of developing diabetes or diagnosing diabetes or monitoring the effectiveness of therapy just using dietary records. We would not dream of doing that. So why are we doing that for exercise? While we know very well that self-reported physical activity might not be reliable to begin with, and also it might not translate to health benefits with the same sensitivity as VO2 max, for example. So maybe we should use more sensitive and reliable markers. Another issue is the relationship between physical activity and cardiorespiratory function. These are definitely related, we know that. So by increasing exercise dose, we can increase cardiorespiratory fitness. But first of all, there are still issues on the determination of physical activity dose. So there is still missing a consensus on how we should actually measure exercise dose. And also, there are other issues. That black box is actually summarized there by a number of factors that affect how physical activity will translate into fitness improvements. So while physical activity is a subjective index, we have still validity and reliability issues. Standard quantification methods are still missing. Is a measure of the stimulus at best and underestimates benefits. On the other hand, cardiorespiratory fitness is definitely objective. It's accurate and reliable. Standards for measurement are available, and it measures the effect, which is what we are really interested in for health purposes. So it is overall a more clinically meaningful measure. So, but how are we going to measure that? So we are used to laboratories and that we can do everything we want there, but in reality, when you switch to the patient and population level, you have to come in a different state of mind. So VO2 max is definitely a very good candidate as a vital sign. Those are basically the reasons why we should be measuring that rather than other things, and I'll go through these one at a time. First of all, VO2 max is an index of global health. In order to bring oxygen from the ambient air and use it up in the mitochondria, oxygen needs to go through a number of conductive and diffusive resistances that limits its maximal flow. So these resistances are represented by the function and the structure of the lungs, the cardiovascular system, the circulation, and the muscle itself. Any changes, perturbations in the structure and function of these apparati will modify VO2 max. So should we have reference normative values? Those curves, those of you who have children will probably recognize some of those curves resemble the growth curves in the children population. I've never actually seen a representation like that for VO2 max, but I think we should have it. So what you see are VO2 max as a function of age in females and males, And those are the different percentile distributions. So should we have those numbers at any time, at any moment in a person's life, we would be able to sort of say where we're standing and to project the person to its future values. So throughout life, the presence of reference data and the periodical assessment of VO2 max will give us a valid marker and the sensitive sentinel of overall current health of individuals. VO2 max is also an independent risk factor for all-cause mortality, for the development of cardiovascular disease, and for the development of other non-communicable diseases, which are, by the way, the main cause of mortality in our countries. So everybody knows about the relationship between all-cause mortality and the quintiles of physical fitness. There is an inverse relationship both in normal subjects and in cardiovascular patients. Probably not as many of you are familiar with the different type of risk that is defined by VO2 max. Nowadays, in surgery departments, people are more and more often measuring VO2 max to stratify patients for their pre-operative risk during general surgery. And even more, they're actually measuring this functional parameter, and also they're envisioning interventions that will increase and pre-rehabilitate the subject prior to surgery. So there is an increasing and enlarging definition of health risk. VO2 max is also an index of the functional reserve, meaning the difference between the maximal functional level and the resting functional level. This parameter influences the independent living, so how many things people can do by themselves without help, and therefore their quality of life, and is also determinant of the exercise dose that the person can assume in a real life condition. If you think of the functional reserve as a blanket, and you consider the energy cost of different forms of activity, and so you have this large blanket that can cover all those activities. Those are the things you can do based on your power. But then if your blanket shrinks, you're going to see a reduction in the amount and type of activities that you can do independently. If you now combine this blanket concept with the well-known decline in VO2 max as a function of aging, you might envision such a thing. And you might also consider this very interesting point. This point is the time, the age at which the functional decline of VO2 max crosses this red horizontal line, which is the amount of oxygen that is required to perform the activities of daily living in an independent manner. Practically, that red arrow indicates when you're going to become non-independent. So that, I think, is a very nice graphic to motivate people to move. So at any given point in time, you can measure a person, and since this decline is really highly predictable, you can project the person later on in their life and predict when they will lose function. So you can also motivate them by saying that at any given moment in time, aerobic training can increase their VO2 max by at least 20 to 30%. And so sort of postpone this coincidence, hopefully after one is dead, so we don't need to deal with it. So there is a complex interaction between VO2 max, exercise tolerance, the amount of exercise dose that I can get, and the health. And all this is like a circle, a complex circle. The amount of exercise or the dose that somebody can have depends upon frequency, intensity, time, and type. We know that very well. Intensity is definitely the most elusive term, and Dr. Keir has been explaining us very well how intensity might in fact be a more tricky issue than just related to VO2 max. So intensity is complicated, especially when you translate it in a real-life condition, because when you have to prescribe exercise that might be performed in a group level, you'll have the numerosity of subjects which will complicate your life. You'll also have the variability in somatotype, the variability in tasks that can be performed, and the variability in the fitness level of the individual, the age, and the sex. So when we think that the adaptation, the positive and negative adaptations of the body are related to specific perturbance of internal cell homeostasis that is mediated by changes in calcium concentration, heat accumulation, the reduction of the ATP to ADP ratio, glucose depletion, energy imbalance, and oxidative stress. These are the main stimuli that will modulate the activity and concentration of PGC1 alpha. It's not like I know too much about this, but I think it makes a very neat connection between real life and how the body functions. So all of this is actually the main mediator of the effects of aerobic exercise on the body, which are increased mitochondrial biogenesis, angiogenesis, and increased fat oxidation, which might well be responsible for the beneficial effects of aerobic training on health. So in simpler terms, these perturbations that mediate all this cascade are related to relative exercise intensity, absolute intensity as well, and exercise duration. So you can see how these different factors can play a very complex role. Actually, defining exercise dose is the first step towards the understanding of the differences in the shape of the positive and negative adaptations to training. This is a graphic that comes from very old guidelines that were issued by ACSM in 1995. And I regretfully have to say that these numbers are still missing. So the general profile is well understood, but real numbers are still missing in the athletic population as much as in clinical populations. And one of the obstacles of the definition of these models is actually exercise dosage. So perhaps the quantification of exercise dose will help us crack the code here and understand what this black box does in terms of the translation of exercise dose in cardiorespiratory fitness. And this is very important. We keep on talking about personalized healthcare. Okay, so this is definitely one step towards personalized exercise prescription. VO2 max is also sensitive to training, which makes it a very good candidate for monitoring the effect of pharmacological and lifestyle interventions. Out of the 1 million graphics I could have used, I've just put on our own work in which you have the changes in VO2 max after 12 weeks of training in a large group of older adults who trained for 12 weeks. These guys trained in two different ways. They trained at the cycle ergometer and at arm ergometer. And they were tested with the same ergometer, with the other ergometer, and this is the control group. As you can see, the relative increase in VO2 max was largest when people were tested with their ergometer with which they trained. So they could achieve about 20% increase in 12 weeks. On the contrary, when they were testing with the non-specific ergometer, the improvement was only about 10%, which gives you an idea of the relative contribution of delivery, oxygen delivery, which is transferable to other forms of exercise, as opposed to the capacity to extract locally. So the reason why we should measure VO2 max as physicians is that this parameter allows us to stratify subjects more correctly, more adequately compared to the value of physical activity. It allows us to make decisions. Some of them are very crucial based on VO2 max. For example, people are put on a transplant list, or we choose different type of therapy or more aggressive diagnostic tools. We can use VO2 max to set goals, specific individual goals, set and quantify training, and to monitor changes. How can we measure VO2 max? Well, definitely we have different options, and what we have to consider is cost as a general term in terms of time, equipment, and available expertise. Effort. Effort can be an issue in certain populations, diseased patients, children, older adults, but also risk. In some conditions, the risk is just unacceptable. And the aim. One very important thing is to remember why we're measuring something, what we have in mind when we need that number. So do we need to make decisions? So we need accurate numbers, or are we just following up? So what we need is like sensitive measures, reliable measures. So depending on what we need to do, we can have different options. So definitely the direct testing, cardiopulmonary exercise testing, is the gold standard. An effort is being made by USA physicians to extensively measure VO2 max in the healthy population in order to generate national norms that will allow us to do a number of things, but definitely give accurate, population-specific, updated reference values. Do we have other options, which might be more practicable at a physician's level? Yes, we do. For example, the same group that you've seen before doing all those extensive measures in a large number in the US are also using those numbers to develop non-exercise equations for VO2 max prediction. So in a very, very large number of subjects, they have used simple equations that are based on sex, age, and either BMI or waist circumference to predict VO2 max. And the performance is quite good, obviously not very accurate, but is a good starting point. So these approaches, non-exercise, they're valuable because they're so easy. They're reliant on measures that we do anyways, so it's very valuable. But, and they can be used to improve individuals' awareness, to identify subjects who are at need for further evaluation. But definitely they have some important defects that we have to bear in mind, which are these indexes are not suitable for short-term evaluation of short-term interventions. So definitely VO2 max could be improving without any changes in either waist circumference or body mass index, and that would not be tracked by these equations. Also, we always have to remember that these equations set their value on the assumption that that subject belongs to that population. Okay, so not necessarily national norms that have been taken in one country, in one specific population, will fit other guys. I don't know if you have ever tried, as Caucasians, to buy a suit in Asia. It just might not fit you because it's built on local physical norms. So I'm not sure, for example, that Northern American norms would apply to the Italian population, just to give you an example. Other options again. Recently, there has been a very interesting work evaluating the validity of estimates, exercise-based estimates of VO2 max that are based on submaximal exercises, and in specific step test, which is a very interesting candidate for the physician's office. So a number of tests have been revised, so there are many options out there. Many of them are actually incremental, meaning they require different level of exercise, so in reality they might not be very, very fast. On average, they represent a high absolute load, so the type of work that is required is normally designed for an adult population and less applicable extensively on an older adult population or a diseased population. The overall standard error of estimate is highest in tests that were developed in very restricted populations, very homogeneous, and the performance is a little less exciting in other populations. On the contrary, the reliability is very high. So these are excellent candidates for test, retest type of designs, so to follow up changes. As I said, there are many options out there, which to me indicates that there's probably not an ideal candidate yet. So one of the reasons, so this is one of the reasons why we undertook evaluation of a different protocol. So I just take as an example the Austrian rhyming step test, which is one of the tests that is most commonly used, has been out there for many, many years. So what are the advantages of this type of test? First of all, is a single trial, and is submaximal intensity, which is normally designed in young subjects to be between 50 and 70% of VO2 max. It's an easy movement pattern. You just have to climb up and down a step. No coordinative challenges too much. It's fast. It takes 5 minutes to do, has a very low cost, and the results are immediate thanks to either nomogram or more modernly equations that can be embedded in a calculation sheet. Why don't we like it that much? Because we often test older adults, and for those people, this test is actually not versatile. The test is characterized by a given stepping frequency and a specific stepping height, which in reality for an older adult population, but even for younger people, but less active than the Scandinavians of the original study, this is a strength challenge for the quads, is definitely an exercise intensity that is too high for older adults. That's the reason why we attempted to modify it. So we proposed about a couple of years ago, with the help of Don as well, a submaximal tailored step test, which retains all the advantages of the original Austrian step test, but we think is more feasible in terms of height that was reduced to reduce the local fatigue of muscles, and the stepping rate has been adjusted just in a similar way as for the Canadian home step test. This is the performance of this test that was evaluated on 60 older adult subjects, healthy. So you have the correlation between predicted and actually measured was challenged against the direct VO2 measure. So you can see data are rather well correlated with an exciting standard error of estimate for an indirect test. So we hope you'll consider this as an option for your future work. So I hope I have convinced you, and that you will be able to convince other people and physicians in particular that VO2 max is an important vital sign. We should forget physical activity and actually measure VO2 max. It's a good index of global health, is an independent risk factor for a number of different situations. It's a good indicator of the functional reserve, which is important for the independence and quality of life, but also is a determinant of exercise dose, and is sensitive to training. So we can use this for mobilizing motivation to lifestyle changes, for goal setting, for personalized exercise prescription, and for monitoring of results. I would like to thank, first of all, Don and Juan for organizing this seminar and making me part of this dream team. I also like to thank CSEP for accepting the seminar and the symposium, and COSMED for sponsoring it. I also have to acknowledge my institutional funding and the people who have been working with me during all these years. So thanks very much.